Okay, let's talk a little bit about the nature of light. Visible light is just one part of the spectrum of electromagnetic energy, or electromagnetic radiation, if you like. And all electromagnetic energy has wavelength. The shorter the wavelength, the higher the energy it has. Wavelength determines what kind of electromagnetic radiation it is, whether it's gamma rays, visible light, X-rays, microwaves, radio waves. The electromagnetic spectrum is the entire thing, from the highest energy, which would be way over beyond the violet end of the spectrum. We would have cosmic rays, then gamma rays, X-rays, ultraviolet light, and then the highest energy visible light, which would be violet. So ultra means above. So ultraviolet is just above the violet end of the visible light spectrum. And then the lowest energy visible light is red. And so infrared. Infra means below. So infrared means below the red end of the spectrum, and that's heat. So lower energy. What we see as the visible light spectrum varies a little bit with different organisms. So some insects and birds can see a little bit in the ultraviolet end of the spectrum. Some organisms can see a little bit into the what we would consider infrared. Now we can detect infrared, just not with our eyes. When you put your hand over a stove burner that was turned off recently, and you can feel that heat on your hand, you're detecting infrared radiation or heat. We just can't see it, but we can detect it with sensors in our skin. So light and the entire electromagnetic spectrum behaves both as a wave and a particle, and the individual particles of light we call photons. So here's our entire electromagnetic spectrum. Cosmic rays aren't on here. They're higher energy even than gamma rays. Cosmic rays come from outer space, very high energy. And visible light is it's one interesting thing to scientists is that the part of the spectrum that we can see, visible light, is also the same part of the spectrum that photosynthetic organisms use for photosynthesis. Why? Why the same part of the spectrum? Why have animals, which don't photosynthesize, evolved to see the same part of the spectrum that photosynthetic organisms evolved to use for photosynthesis? And one possible reason is that this is the most energetic part of the spectrum that doesn't damage DNA. So visible light and anything lower in energy, infrared, microwave radiation, radio waves, these are not high enough energy to damage DNA. Ultraviolet is. And the higher energy it is here, the more easily it penetrates the body as well. So ultraviolet light can damage DNA, but it doesn't penetrate very far into our body. So ultraviolet rays from the sun can cause skin cancer by damaging your DNA, but they're not going to cause liver cancer or brain cancer because they simply can't penetrate the body like that. X-rays penetrate much better, but they penetrate soft tissue much more easily than bone and calcified tissue. So that's why X-rays are so useful for medical diagnostics, because they penetrate some types of body tissue more easily than others. Gamma rays rip right through everything in our body. They go through bones and bounce around off of things in our bodies and are much higher energy. So good for killing cells, not so good for diagnostics. And anything down here is not energetic enough to damage DNA and also doesn't penetrate the body. So microwaves, which is what cell phones use, which again recently there's been some nonsense about new 5G technology is going to cause brain cancer or something. Microwaves are simply not energetic enough to cause cancer by any mechanism that we are aware of. They're very low energy. They don't penetrate the body. how are they going to cause cancer if they can't even get into your living cells because it's too low energy? So the visible lights, what we call visible light, is what humans see, which is what mammals see. And the parts of this spectrum that are used for photosynthesis are both the violet and blue end of the spectrum and also the red end of the spectrum. Green is not used for photosynthesis. Green light is right in the middle of the spectrum of visible light, and chlorophyll doesn't absorb the energy from green light. Green light just bounces off of it or goes through it, and that's why most photosynthetic organisms look some version of green, either really bright green like plants or kind of a brownish green like a lot of protists, because they just simply don't use green light. And what a surface absorbs is not the color that we see. What they reflect is the color that we see. So when light hits chlorophyll, both the red end and the blue end of the spectrum are used for photosynthesis, but the green and also mostly the yellow part of the spectrum are not used for photosynthesis. And they are just transmitted right through or reflected off, and that's why things that are dense with chlorophyll look green. So we'll talk about the two main kinds of chlorophylls in plants, chlorophyll A and chlorophyll B. They have both an absorption spectrum, which is exactly which parts of that spectrum of visible light those chlorophylls absorb, and also how well those different parts of the spectrum drive photosynthesis, and that's the action spectrum. Sometimes the amount of oxygen that's produced is used as a measure of how well different parts of the spectrum drive photosynthesis. So chlorophyll A and chlorophyll B, which are similar but have slightly different absorption spectra. So if we shine all the different wavelengths of light through these pigments and then measure how much is absorbed, we find that for chlorophyll A, it absorbs slightly more energetic light than chlorophyll B. And so if you can actually separate the two chlorophylls if you remove them from plants, extract them using organic solvents. And chlorophyll A is a little bit more teal colored and chlorophyll B is a little bit more olive green in color. And then there are these other pigments, carotenoids and xanthophylls, that are not greenish in color but are oranges and yellows in color. And it used to be thought that these participated somehow in photosynthesis, but now we know that they don't actually participate directly in photosynthesis. The energy and the light they absorb is protective to the chloroplast, but it doesn't directly participate in photosynthesis. So that's the absorption spectra. The action spectrum is how well these wavelengths of light drive photosynthesis. So you can see we have lots of oxygen being produced here by wavelengths in the violet and the blue, and also on the red end of the spectra, and almost nothing in the green and the yellow part of the spectra. This was discovered over 100 years ago by this guy Engelmann and his classic experiment using an aquatic plant, Spirogyra, to determine how well different parts of the spectrum drove photosynthesis. And because back then they didn't have any way to measure oxygen production directly, he used the growth of a bacteria that required oxygen as an indirect measure of how much oxygen was present. And he actually did a really good job using that measure, determining what part of the spectrum was better for driving photosynthesis. And using a simple prism to separate light to shine on his aquatic organism. So chlorophyll A is the main photosynthetic pigment. Chlorophyll B is considered an accessory pigment. It helps bounce high energy electrons around. And then the carotenoids absorb excessive light. They help prevent damage to the chlorophylls, but they don't participate directly in photosynthesis. So this is what chlorophyll looks like. If you remember, chlorophyll is considered a lipid. It's not very water soluble by itself. We have to use organic solvents to extract it. It has this long carbon hydrogen tail that allows it to stick in the membrane. Remember, things stick in the membrane just by hydrophobic interactions. So chlorophyll is one of those. Because it has a long fatty tail, it just sticks in the fatty portion of the membrane of the thylakoids just by hydrophobic interactions. And it has a porphyrin ring. We've encountered another porphyrin ring, and that was in hemoglobin. Hemoglobin has, each globin subunit has a porphyrin ring, but instead of magnesium in the center, it has an iron atom in the center that helps it carry oxygen. So porphyrin rings typically have a metal in the center. So chlorophyll has magnesium in it, which is one of the reasons why green plants are rich in magnesium as a source of magnesium, because chlorophyll always has magnesium associated with it. Chlorophyll A and chlorophyll B are almost identical, with the exception of one functional group here. So what chlorophyll does, so chlorophyll is really the magic molecule in photosynthesis. What it does is it takes light and it allows electrons to bounce up to a higher energy state using light. And that's really the step one of photosynthesis, is turning the energy of light, photons, into excited electrons that can then be passed on to eventually be used to make ATP, or to be passed on to an electron carrier for the synthesis part of photosynthesis. If there's nowhere for this electron to go, eventually the electrons will drop back down to their ground state and release their energy. And we can observe this in a simple lab experiment, which if you go on to take biochemistry or a botany course that has a lab, you might get to do this experiment. I remember doing this experiment eons ago. You extract chlorophyll from plants using an organic solvent like acetone, and you expose it to light, and this will allow all the electrons that can to bounce up to a higher energy level. And then you put it in the dark. And because those electrons are dropping down in energy level, they will release their energy, and it releases a specific wavelength of light and also heat. So it will warm up as those electrons go back down to their ground state and release their energy. So this is a good demonstration of how in both of these processes, respiration and also in photosynthesis, we're not creating new energy. We're changing energy from one form to another at every step of the process. So photosystems are in the thylakoid membrane. This is where chlorophyll is. This is where it's a large complex containing many polypeptide chains and also embedded chlorophyll molecules. And this is a nice ribbon structure of one of these little dotted lines. That's the edge of the membrane. And this whole structure here is one photosystem. And the little purple things in here, those are the chlorophylls. I don't know why they made chlorophyll purple in this one instead of green. I have another image of one where it's green. But these are where those initial reactions are going to take place. And there's two different types of photosystem. One that produces oxygen as a byproduct and one that does not. And they behave slightly differently when it comes to photosynthesis. So in general, how a photosystem works. So photosystem is what I just showed you. I'm going to go back to the previous slide. So that's what it actually looks like. But this is a little bit more simplified version of what a photosystem looks like. So they're embedded in the thylakoid membrane. So you can see the membrane here. And they contain chlorophyll. So photons hit the chlorophyll molecules, and excited electrons can then start moving around and being transferred from molecule to molecule between the chlorophylls until eventually they reach the reaction center of the molecule. And the reaction center is a pair of special chlorophyll A's that are slightly different. Again, we don't have to worry about exactly how. and they're going to allow the electron to move on, to be transferred out of the complex here to be used for another purpose, either to an electron transport chain or an electron carrier, one of those two things. So that's what's happening in both types of photosystem. And we call them Photosystem I and II. You notice we use Roman numerals here. And initially they were named Photosystem I and II based on the order of their discovery. So Photosystem I was discovered first, Photosystem II was second. But it actually turns out that's also the order, just coincidentally, that they evolved. Photosystem I is the older photosystem, doesn't produce oxygen as a byproduct. And Photosystem II is the one cyanobacteria have and the one all the plastids have. Well, they actually have both. But this one evolved second. So the primary electron acceptor gets reduced, and it allows these high energy electrons to be transferred, and that's really what the light reactions are doing. They're using light energy to turn it into high energy electron and then pass that on so that it can be used for something else. So Photosystem II, we'll talk about that first. In plants, it handles lower energy electrons, so that's the first one. So we'll talk about that one first. It has a reaction center that has two chlorophyll A's that are called the P680 chlorophylls, because if you measure which wavelength of light they absorb best, it's 680 nanometers, which is in the red end of the spectrum. So here's another image of one of those, a ribbon model of one of these photosystems. So the little green structures here now are chlorophylls, and the rest are different polypeptides that are complex together to form this structure. And so we have the inside of the thylakoid in here, and the stroma of the chloroplast is up here. So Photosystem II. That's the structure. Photosystem I also has two chlorophyll A's at the reaction center, that where the electron is going to be passed on to an electron carrier. They absorb best at a slightly different wavelength, so we call them the P700 chlorophylls because they absorb best 700 nanometers wavelength of light. And they have a slightly different mechanism of handling the electrons. Photosystem I can actually bounce electrons up to a higher energy level.